Good morning, students. Today's topic is semen analysis, and another topic is effect of removal of gonads. So we'll start with semen analysis, followed by the removal of gonads. Now, coming to semen analysis, semen consists of spermatozoa that suspended in plasma-like fluid and is formed at ejaculation. That we have these things we have seen in the previous class, how semen is formed and uh, what part uh, what part of it is fluid and we have sperms. So, what is the contribution of which gland? All these things. So only the spermatozoa and a small amount of secretions are produced in the testis and the epididymis. Bulk of the semen consists of the secretions of seminal vesicles, that is and the prostate. uh, the As we have already discussed, the secretions of seminal vesicles, they amount to around nearly 60% of the entire volume of the semen. Bulbourethral and urethral glands contribute to around less than 1%, as we already seen. So initially the sperms are not motile, but while passing through the epididymis, they acquire motility by virtue of carnitine that is added to the seminal fluid in epididymis. So once these sperms they travel from the reach testis, as we have already seen, from the seminal, uh, from the reed testis to the uh, epididymis, they stay for a few hours, around 18 to 24 hours. In that period, hitherto they were immotile. Now they acquire motility by virtue of carnitine that is added to the seminal fluid by epididymis. Now, sperm analysis. Uh, what do we do? Seminal vesicles provide an important energy source in the form of fructose that enhances motility of spermatozoa. Prostaglandins and fibrinogen-like substances are also added to seminal fluid by seminal vesicles. We already seen prostaglandins. They help in changes, bringing about changes in the cervical secretion, so that sperms can move through. Right, that is one action, and they also stimulate antiperistaltic movements in the uterus and fallopian tubes, so that sperm can be directed towards the ovary. So, and fibrinogen-like substances are also present in the fluid secreted by seminal vesicles. And like that, prostatic fluid also provides a number of enzymes, some ions like citrate, calcium, and zinc. The bulbourethral and urethral glands contribute to mucoproteins and IgA to semen. Right. So we can see the diagrams. On the right, you can see spermatids undergoing differentiation, Sertoli cells, spermatogonium. All the, it is depicting the stages of spermatogenesis. Now, when is semen analysis required? That is called indications. What are the indications for semen analysis? These include infertility, hypogonadism, follow-up after vasectomy, prior to donations for artificial insemination, and storage of semen before radiotherapy. Right. So when there is infertility, to check whether the infertility is because of male infertility or female. So as part of that, you need semen analysis. And hypogonadism, the gonads are not grown and developed to the normal extent in that case also. And follow up after vasectomy. After vasectomy is done, whether it has been successful or not, whether the sperm count is completely totally absent, or still you can see sperms in the semen, it shows the success or otherwise of the vasectomy. Now, prior to donations for artificial insemination and storage of semen before radiotherapy. So, in patients with certain cancers, the radiotherapy is given, which affects the spermatogenesis later on. So that is, in that cases, prior to the radiotherapy itself, semen is collected and it is stored before radiotherapy itself. So like that. How is the sample collected? A period of abstinence is important as it affects both the quantity and motility of spermatozoa. So there should be ordinarily at least 3 to 5 days of abstinence, sexual abstinence. So because the quantity and motility of spermatozoa should not be affected. The specimen should be collected in the morning to allow sufficient time for its analysis. Masturbation is the ideal method for producing the semen specimen. Condoms must never be used for collection of semen by intercourse. Okay, so note the volume, color, appearance, and pH. We have to examine volume of the semen, color, appearance, and the pH. Now, a clean, dry, wide mouth glass or plastic jar should be used as semen container. Its lid must not be rubber lined. Detergent, water, and rubber are injurious to sperm. These are certain precautions while collecting the semen. Specimen should be transported to the laboratory at a temperature as close to 37 degrees as possible and delivered to the laboratory in less than 2 hours. This is very important. Temperature maintenance, because as we already seen, the sperm, the viability of sperms is they're highly sensitive to temperature changes. So that is why it is mentioned. So if it has to be transported to the laboratory, there should be a close maintenance of temperature at around 37 degrees, right? Now, normally the human semen soon after ejaculation forms a gel-like clot that liquefies in 5 to 20 minutes. Absence of liquefaction in a semen sample must be noted. So we have already seen there are uh, certain kinases in the seminal vesicle fluid, which causes which are acted upon there are certain secretions in the prostate gland secretions which cause the coagulation kind of activity in this semen. And until the semen has reached the near the cervix, once it is reached near the cervix, after few minutes, what happens is this profibrinolysin, it gets activated to fibrinolysin, which causes which acts on the already, I mean, clogged coagulum kind of formed semen. It is acted upon by the fibrinolysin. So finally what happens, this coagulum will be liquefied. That means the clot like structure, the jelly like structure will be liquefied. That is what we have to observe, whether the liquefaction is happening or not. Because if this is not happening, it will not be helpful for the sperm to penetrate properly through the cervix. So it is also a very important step. Now, viscosity of semen should be assessed. It can be measured by dropping a drop of semen from a 10 centimeter long capillary tube containing 0.1 ml semen. Uh, Time taken by the drop to form and leave the capillary tube is a measure of its viscosity. Uh, Time taken by the drop to form and leave the capillary tube is a measure of its viscosity. Quite understandable. Now coming to sperm count. First of all, there should be visual assessment. Place a drop of semen on a clean glass slide. Place a cover slip over it. Examine the slide under the high power objective of a microscope. Make a visual assessment of the sperm count and to determine the need for any dilution. So in case of requirement of dilution, diluent should be used. These are the intricate details of the exact laboratory methods of sperm counting. Now the counting procedure, like you have already seen, the hemocytometer Newbar's chamber is used, and the WBC squares are used, as you can see. Okay, the chamber is then examined under 10x objective microscope. These are the calculation part. Now, like we already discussed, not only number the sperm count not only the sperm count is important, along with that motility is also very important. So assessment of motility must be performed soon after production of sample, 3 and 6 hours later. Both motile and immotile sperms are counted at least in 5 fields with a minimum count of 200. The count should be performed in duplicate and the average recorded. Only forward movement of sperms is taken as positive. Percentage motility is then calculated. So first of all, motility, motile sperms, what percentage are motile sperms and immotile sperms? In that motile sperms, only forward moving sperms are taken as positive count. So that is something about motility. The sperm count can be calculated by the formula motile sperm count is equal to sperm count by ml percentage. More objective results can be obtained by following procedure. These exact procedures are given. You can go through the slide. Now there is grading according to the motility, like from grade ranging from grade 0 to grade 4. Grade 0 is no movement at all. Grade 1 moving with no forward progression. Grade 2 is moving with slow and wandering movement. Grade 3 is moving rapidly in almost straight line. Grade 4 is moving with high speed in straight line. That comes to normal. Motility score, there is a procedure for grading and motility scores. Normal motility score for spermatozoa is greater than or equal to 150, right? Now, how are they reported? Some of the special terms used for reporting the results of semen analysis are aspermia, means there is no ejaculate itself. Oligospermia or hypospermia is reduction in volume of ejaculate. The volume of the ejaculate is less. Semen volume itself is less. It is called oligospermia. Hyperspermia, increase in volume of ejaculate. Oligozoospermia. So be careful. Oligozoospermia is low sperm count, that is less than 30 million per milliliter. Polyzoospermia, higher sperm count, that is greater than 300 million per milliliter. Asthenozoospermia, absence or marked reduction in sperm motility, which is motility score is less than 150. Necrospermia is dead sperm. So these are certain terminologies used in reporting the sperm analysis, semen analysis. Assessment of sperm morphology also. A normal sperm consists of a head and a tail joined together by a short neck. The head is oval in shape and measures 4.5 by 3 by 1.5 micron, while the tail is about 50 micron long. Assessment of morphology can be made in a wet preparation or in a stained smear of semen. The slides are then examined under oil immersion objective of the microscope. Abnormalities of head include small head, large head, tapering head, pyriform head, amorphous and double heads. All these are abnormal shapes of head which cause ineffective sperms. That means the motility will be affected. The tail including double, coiled or short tails and of mid pieces should be noted. All these are abnormal morphology. They should be noted. Uh, this is a reported, reporting terminology is repeated. Uh, this we already seen, it is repeat. Now see, you can see the normal sperm form and abnormal sperm forms, double headed, double tails, like that, different morphology of heads, all this. So you can see the abnormal sperms. Now reference range of normal semen. Normal semen should be around the range should be 1 to 10 ml. Color is gray yellowish. Appearance, it appears opalescent. It is viscous and the pH ranges between 7.2 and 8.9. Average will be around at least 7.5 alkaline. Now sperm count is normal range will be 60 to 150 million per ml. Some extreme range will be 30 to 300 million per ml. That means anything over 300 will be abnormally high, right? So motility greater than 70% at 1 hour and 50% at 3 hours after ejaculation uh, will be taken as normal motility. Motility score should be greater than or equal to 150. Morphology should be greater than 70%, should be morphologically normal. That means greater than 70% of the sperms should be morphologically normal. Now, one more important thing, testing for anti-sperm antibodies. Testing for anti-sperm antibodies is as important in the evolution of infantile male. Um, it is important in the evolution of infantile male. Infertile male. Uh, Agglutination tests, sperm immobilizing antibody tests, testings for cytotoxic antibodies are the various methods for demonstrating sperm antibodies. So, in infertility, along with all these morphological examinations, anti-sperm antibody examination is also important. Types are agglutination tests, sperm immobilizing antibody tests, cytotoxic antibody tests. Some miscellaneous topics. So, certain important points. An important cause of aspermia is retrograde ejaculation. So, aspermia means no semen at all. So, it is retrograde ejaculation backwards into urinary bladder. In all cases, when ejaculate is not obtained, a urine specimen should be immediately collected and examined for spermatozoa. So, if the urine sample which is immediately collected, if it shows sperms, it is an indication that there is retrograde ejaculation happening. An immotile sperm does not necessarily mean a dead sperm. It is important to distinguish between actinozoospermia and necrospermia. For evaluating infertility, semen analysis should be performed on 3 occasions with a gap of 2 to 3 weeks between any two analyses. For evaluating infertility, important. Semen analysis should be performed on 3 occasions with a gap of 2 to 3 weeks between any two analyses. That means, what do you understand? That means the semen, the characteristics, the volume, counts, etc., are highly variable. That means it changes according to, I mean, on different days it can be different. So you cannot take one sample and one report, and it cannot be, you cannot say it as diagnostic. That is why to neutralize all these, equalize and neutralize all these variations, semen analysis should be performed on 3 occasions with a gap of 2 to 3 weeks between any two analysis, right? So if today is the first semen analysis, after 2 to 3 weeks another semen analysis should be done, and later again after 2 to 3 weeks another is done. So on 3 occasions it should be done. So this is about almost about sperm analysis and semen analysis. Now we'll continue with another topic, effects of removal of gonads.